Okay, so here we are with our part 2 of the So we left off with the whole idea between natural or artificial or acquired, right? And then we can break down the acquired into natural or artificial, right? And then with that, that's going to tell us how we're exposed, is how we acquire something. So I got a good graphic in this PowerPoint to pick up on that. But we'll just get through some. That's where we left off. So when we come to that, we'll expand on that even more. But we got to get through this preliminary stuff just to make sure we have it all tightened up, because we missed out on that being so clear in the recording last time. And make sure that video is not doing anything. Okay, good. All right. So the first part of this part 2 PowerPoint is really kind of tailing in from where we left off on the other lymphatic PowerPoint from last week. But this PowerPoint is going to be much more specific on the adaptive part of the system. In our last PowerPoint, we talked a lot about the innate systems, which included fever, with pyrogens, those proteins that mediate a fever. And then we saw interferons helping with viral infections, where they're released from a virus infected cell. That viral infected cell will release these interferons, which will basically warn and arm other cells, other neighboring cells, to produce these same antiviral proteins that will kill or at least denature viruses to some degree. And then we saw a little bit of what an antigen presenting cell will do. So that's what we can expect to see in this first part, and then we'll get to the more adaptive stuff. So remember, adaptive, this is specific. This is third line of defense. Everything we did last time was first and second line. Again, this is a repeat. So if you're saying, why is he doing this again? It's because the last video got all messed up. So first line is basically your skin barrier. Anything that comes off of your skin barrier or any other lining barrier. So you having mucus or sweat or tears, anything that washes away the external barrier or the internal barriers would be still part of the first line of defense as a barrier. And that would include that, what's that called? That acid mantle. Remember hearing that one before? That protects even more for the lower pH, because certain bacteria don't like that. And the second line of defense, that's where we get a little fancier with inflammation. So redness, heat, pain, swelling. Remember those 4 cardinal signs? Second line of defense. So inflammation is cardinal, like cardinal up top of second line defense. Then we have fever as part of that. We have macrophages doing their thing all day, all day, all day. That's part of second line. Even natural killer cells, even though natural killer can be part of second and third line of defense, natural killers can be non-specific, right? So they're still part of second line defense, or they could be specific, which would be third line defense. Anytime you get to a specific antigen being targeted, that's third line. In the case of natural killers who have enzymes to kill, they can be both. So that's what we'll see. But anytime you get to real specificity with antibody production for a certain antigen, that's all B cell adaptive defense. And we'll learn that B cells, as great as they are in a sense, you could almost say useless without the T helper cells. So there's the T helper that kind of like is the party planner, right? It's what sets the stage for things to happen correctly with our whole B cell response. So if you want B cells to do it, to do the best work, you need a T helper to coordinate the B cells to do the best work. So there's that tandem there. This is a nice slide we saw last time, just showing you a little bit of preview. So test, question, objective right here. What is first? What is second? What is third line? There you go. So screenshot that. Just know to come back to this slide and know what's what. Also know the keywords here. Know that B cell are part of what's called humoral immunity, and that T cells offer what's called cell-mediated immunity. It's B cell for humoral because the antibodies are released into the humor, which is the bloodstream, which go all around the body. Again, anything adaptive third line is always systemic, which means a full body response, right? T cells offer cellular immunity with cell-mediated immunity, I should say, because we need a APC, which is an antigen-presenting cell, to basically activate the T cell. So here you go. You can have a pathogen over there and a T cell, the bad guy that they are, right? The good bad guys. They're gonna look at that antigen, they're gonna recognize it. But they're so cool, they're so up on the chain, they're not gonna touch that antigen themselves. They're gonna say, a little APC over there, you go introduce yourself, tell them they're about to meet their demise, right? So the T cell is not going to go straight out right. It's going to let an APC bind first, and the T cell is going to come in and kill it, okay? So they need another cell to get them engaged, is what I'm trying to say, right? That's why they're cell-mediated. So the more you feel comfortable with the terminology, that's why, man, I remember when I was first learning this way back, you try to get all this vocabulary down, and then you go in for the kill to try to learn all the mechanisms, and you get lost. When you feel lost, it means that you need to come back to do the vocabulary again. If you know the terms well enough in the very beginning, you start to really unfold the chapter and you get to understand the mechanisms easier. And that's something that was drilled to me way back from my first big physiology teacher. At the beginning of every chapter or unit, whatever it would have been, he gave us a whole list. I felt like there was a little bit too much. 100 to 200 terms. I'm like, good God, and I would wonder why? Well, now I know why. If you don't know the language, you're not going to be able to speak it. So you got to know the lingo. And I've seen many people. It's been a while since I taught A&P, but that's what I did for a long time, for like 6 or 8 years straight. People miss stuff like that. They miss the coordination between B cells being humoral immunity. With B cells come plasma cells, with plasma cells come antibodies. You got to know what words belong to what system, right? T cells have helper cells and cytotoxic cells. Do you know that, or do you not know that? Right? B cells will not be cytotoxic, right? And T cells will not give antibodies. So that's how you miss test questions, by not being able to line everything up on the front end. So again, it's going to be B cells that give us antibodies, and they don't kill outright. Antibodies just kind of like confuse pathogens, right? They got a lot of busy work to keep the pathogens away from either spreading or getting stronger. So B cells are kind of like distracting to pathogens. It's the T cells that kill, okay? So we'll see that. So we're going to blow through these. This is first line, our skin, acid mantle right there. Anything of helping that barrier for first line. Second line, we got macrophages, all the things we learned before, eosinophils, neutrophils. That's all part of your second line for phagocytosis. Anything that's non-specific, right? Just a regular old pathogen floating through your body, getting engulfed. is second line of defense. As soon as that's a specific antigen that needs to be bound to activate a B or T cell, that's when it becomes specific. So we looked at these terms last time: chemotaxis, adherence, ingestion, digestion, egestion. This is simply the pathway that a phagocytosis is going to take. So in this picture, we see chemotaxis is what attracts a phagocyte to a specific area. And you can remember that because taxi kind of means to move, I guess you could say. Chemo means it's a chemical attraction. So anytime there's a danger in the body, there's going to be a chemical trail. There's always a chemical trail to pathogens moving around. So these macrophages are going to know that chemical trail. That's what's going to attract them. Next picture shows adherence, which means it's going to grab onto that pathogen here in the blue. It's going to engulf it, digest it, and then egestion down here. So all the steps you see here is what's going on in that picture. And truthfully, that's kind of high school. It's not that hard to. I mean, it would be a lot to memorize all that. But if you just think about the story of it, if you just close your eyes, you can imagine a phagocyte, being attracted to a pathogen from the chemical trail, ingesting it, digesting it, and then egesting any of the waste. And sometimes this waste is in the form of a protein fragment. That microbe, if it's acting as an APC, as the phagocyte, if it's acting as an APC, will display a piece of that digested protein on the surface of its cell, which then will attract a T cell to come around and bind to it and activate it. You're not going to get any activation until something is bound. And then we'll see through different mechanisms that T cell, once it's activated, can start spitting out these other chemicals that will attract more B cells to be activated or more T cells to be activated. So it's a very, together effort. Nothing really happens on its own in the immune system. It's a very much a group effort. So the act of moving across this phagocyte would be an example of transcytosis. Another second line of defense here, this natural killer, releasing perforins and then injecting these chemicals. Cytolytic, if you guys don't know that term, cyto means cell, lytic refers to lysis. Y'all remember that in AMP1, what it means for a cell to swell and blow up, right? So that's what these natural killer cells can do. Then interference, like we said, the original cell will be infected and die, but before it dies, it's going to release information, right? Like a good spy would, right? Like in those movies, and send over the data right before they die. Sounds like a good Star Wars movie, doesn't it? Isn't that what Princess Leia and R2-D2 pulled off? Yep.
One of the questions that I remember on the test is where does red blood cells die?
Yeah, spleen. Yep. That's the graveyard. Yeah. Well, here we go. Yeah, if you haven't seen the last 5 minutes of Rogue One in the Star Wars series, you got to see the last 5 minutes of Rogue One. Holy Jesus. That's like excitation of every cell in your body if you're a Star Wars fan, right? Anyway, that's kind of what's going on here with the interferons. They can give antiviral proteins to other neighboring cells to know how to stop viral infection. Isn't that cool? Complement system, as little as these little boogers are, these are little proteins. They represent a huge, vast across the board, more like a general immunity. They're not really specific. They're still second line, but they sure have a strong impact upon the whole immune effort. So yeah, a major mechanism for destroying foreign substances in the body And they do this by tagging certain pathogens using opsonins. These are little protein markers. So they're not going to like pick on certain pathogens. They're going to look at anything that's non-self, and just tag it. We put a little protein on it, and something later will come and eat it because it'll be tagged for death, you know? That's what an opsonin is, a protein marker for death. It's a very concerted effort, like I said before. And this is what, you got to recognize, and I guess the better word for it is respect. It really is about respect, because the immune system often is disrespected. People think, and I don't want to cause any, arguments or nothing like that, but people think that immunity comes from medication. It does not come from medication. It actually comes from the lack of medication. In order to build a healthy immunity, now for people that are gravely ill, yeah, you need medication to stay alive, absolutely. But the regular everyday person without any, underlying serious illness, the best chance they've got is exposure to build a healthy immune system. And that is the unique challenge that COVID brought, because we were forced, for some good reason and some not so good reasons, to, mask ourselves off to the whole world, which helped in some ways, but not in all ways. We lost immunity for like 2 years. The whole world was masked up, and we lost that general everyday exposure, which builds your immunity every day. And for young people, we got over that fast, but for elderly, they don't come out of that really quickly. When you face off an elderly person from everyday exposure, yeah, it could have saved their life from a viral infection, I suppose, but it also, sabotaged your immune system, which meant then they could get something small like a cold and then get COVID on a snap like that, because your body has no way to fight it. So it is kind of sad in both respects. It's tough. There's our inflammation. I don't want to spend much time on that, but all these have a purpose, guys. It's all about increasing circulation, swelling to increase permeability of immune cells coming in to heal and to take away. So read through these guys one by one. I think of in the new PowerPoint, I've got certain things reddened or bolded that you need to pay attention to. So just come back and view those. I don't want to spend time on that. Getting to our third line, finally, we did bring this up last time. The 3 hallmarks being specific, systemic, and memory. Memory is the cool thing about third line. What this really means in memory is that on a second exposure to the same antigen, we have a much faster response. That's what that really means. So you might get a question of which of the following passages would indicate a memory of immunity. And you want to look for future exposure or second exposure having a faster turnaround, right? And we'll see a graph that helps us, understand that. Graphs are good in that way. Are we good so far? Everything making general sense? Okay. That's how it needs to be. Because I know when you study this stuff, it seems just like a parade of terms. You need to think of it more so as a story. If it can make sense to you, here's how to study in a better way. If you're reading something in the PowerPoint, or maybe rewatching the video, whatever, pause it, stop and think about it, internalize it, how you would explain it to someone. If you can do that, then you got it. If you can't explain it to someone, then it means you probably don't have it. So if you are one that watches these videos again, that's the best thing, is maybe just watch 15 minutes of it, and then sit and ponder in between all the dumb jokes that I might say. Think if you could explain it to someone, or maybe jot down your own notes in your own words. Guys, that's actually how I came to understand any of this, is I never could get it from the professors, ever. They spoke a whole different language than me, it seems. and they were happy to do it. They were not in any prioritized hurry to make sense to anyone. That's how education, I think, used to be in college. Maybe it still is at universities. But yeah, it took me just finally just getting mad enough to understanding it on my terms, I guess, and that's how it seemed to help. So maybe that's the strategy for you. So if you're not getting it from me, I'm begging you to please watch those videos from Miss Meyer that are posted. She might be a better communicator to you, and that's perfectly fine. I'm never hurt by that. Trust me. Talk to the tutors, talk to a different friend if it takes. That's how it goes. All right, so here we are with the types of immunity. So again, a simple concept, but if you don't play with this enough in your head, it can mess with you on the test. So when we're talking natural and artificial, that's how it's acquired. Okay? That's how you get the pathogen in the body. So if you naturally are exposed to a pathogen, it means you walk by somebody, they coughed on you, or you touched the doorknob and you put your hand in your mouth, or you ate some nuts from a bar, like along came Polly or something. That's natural, right? All natural. Artificial acquired would be you getting a vaccine, pretty much, right? Something, the pathogen, which you normally wouldn't do, but in this case, a vaccine, the technology behind that, you're given the pathogen so that you can build immunity to it, hopefully. And these are done with the traditional vaccines when the pathogen is what's called attenuated, which means it's less powerful. It's been dumbed down, basically, diluted down or broken down to where it's still there, but not in its full force, so that when it's introduced to your body, your immune cells can jump on it before it has a chance to jump on you. So it's like it's asleep. Think of it like that. When you talk active passive, that means what's your body going to do? So natural artificial is how you're exposed. Active passive is what did your body do with that exposure? Did it produce antibodies, or were you given antibodies if your immune system is shot and you can't build antibodies? So the passive side of immunity is going to be very, very rare. The only really examples of passive immunity is breast milk, which is passive antibodies given to the newborn from the mother. That'd be one of your shining examples of passive immunity, because we're being given antibodies, right from the first point of being breastfed, the baby is enriched with these beautiful antibodies that have been passed down for generations, so that's wonderful passive immunity right there. The other indication would be you go to a hospital and they inject you with antibodies. And that would really be if you're just, somehow immunocompromised where you weren't able to build your own. Otherwise, active immunity is going to be when you build your own antibodies, which would happen with a vaccine. It would. That's what artificial active immunity would be. Getting a vaccine, artificially being injected with the pathogen, right? You building immunity to it, active immunity. So please know that example of artificial acquired active. That's your typical vaccine-induced building antibodies on your own, right? That's the beautiful part of vaccines right there. That's what they're designed to do, which is a beautiful science. It's just, it'd be nice if those vaccines were not so screwed up in the process. Some of these are not purified as they need to be, and they're introducing monkey, rat, and cow and pig DNA into our bodies, which is not so cool. For most people, not a problem. Honestly, that's why they get away with it. Like all drugs, for most people, no reactions, right? For some people, there is a reaction. Some people, this produces cancer. Having these exogenous proteins from monkeys and rats being introduced to your body, sometimes that's a problem for people. So it's almost, you could say careless for the pharmaceutical companies to do that, but It's done in the same light that all drugs are done. It's a percentage of population of reaction. If the percentage is low enough, they consider it safe. So I say that just to give you the full story on vaccines. I'm not trying to teach anti-vax at all. I just like for you to be informed, that's all. To say that you don't want to be informed would be silly in the medical community, honestly. It's irresponsible to not be informed. So at least now you're informed. That vaccines are meant to be good, but sometimes they're not good for all people. So you getting the chickenpox from 3rd grade would be natural active, right? Natural exposed, and you built your own antibodies. There you go. So I thought it would be best to just run through those. And you can re-listen to the video if you need to. Rather than reading all the examples, it might be easier to hear them. So anything with passive immunity, again, would be breast milk. Like natural passive would be your mother giving you breast milk. Artificial passive would be you were given a vaccine and you still couldn't produce antibodies. So they got to give them to you in the hospital. All right, so we can skip all this now that we've talked about all the different ways. Same with this, just read this on your own, let it make sense to you. Maybe jot it down in your own terms, but everything on here is what I just explained. Any questions before I move on from that? Okay. So yeah, the same confusion like blood typing, you know, understanding what an antigen would call out the blood type is a simple concept to learn, but when you hear it on the test, somehow it washes all kinds of fuzziness across your brain. This can do the same thing. It's an easy concept. You got to practice it so you don't get fuzzy on it for the test. Again, active passive is everything to do about you building antibodies or not. Active is building antibodies. Passive means you're not building antibodies. You're being given the antibodies. Which would be in the plasma, by the way, the serum. It's not on the red blood cell. This is the serum. This is the plasma.
Active immunity, building antibodies, and you said passive immunity is being given.
Perfect. Perfectly stated. Yep. That what you did right there is how you learn it. Exactly. You say it over and over again. You nod your head when you're saying it, that it makes sense. You go write it somewhere, you email it to yourself, you tell someone else, whatever it takes. Then you don't have to study it. If you store it correctly in the brain, I promise you, you'll find yourself less stressed when studying. It's like walking into a closet of a bunch of beautifully folded towels. They're just sitting there waiting for you, right? If you store away that information correctly in your brain, it'll be there waiting for you for a test. If you just throw it in the closet, a whole different story. So a lot of times we just throw information in our brain and we don't go back and tend to it. That's what makes our brain all kinds of messy. And that's why I always laugh that I am a teacher, because my brain was messy for a mighty long time until I decided to, I swear I got so mad, basically crying, cursing God, you know, come on now. I need to get this stuff, and finally sat down and did it for myself. And it stuck, you know, what can I say? I don't know everything, but I know, I guess, enough to get by. So anyway, antigens are going to be anything that's non-self. In the case of red blood cell antigens, those are specifically called self-antigens. Okay, so we're going to ignore that. A little bit of confusion there. Red blood cell antigens are self-antigens. Those are okay. But everything other than that is going to be considered a non-self pathogen. So they're also called immunogens. Antigens are also called immunogens. Because anything gen is what starts something. So this is going to start your immune system. It's like a Kickstarter. They start an immune response. That's why they're called immunogens. So you got to wonder, why did God make so many antigens to screw with our body? Yeah, I don't know. I guess it's the dance that we all go through.
You said it starts what?
An immune response. Yep. Why must we go through life constantly being provoked by certain proteins? Why must they be there? I mean, that's a good question, right, to ask. I guess it's it's just the human condition of constantly learning and building and growing from whatever is bad turning good. You know, who knows? It's too deep. Who knows? This is the way it is. So, you know, I have a whole different take. And again, I'm not an anti-vax person. I'm not. Anti-stupidity, yeah. But, you know, you got to realize you cannot just get away with a statement like, we're going to produce a vaccine for every virus. That's just not going to happen. You know, you're always going to be chasing mutations of viruses for thousands of years. You're never going to get something where you can completely contain a virus with no questions asked. Viruses are damn smart, and they always will be. So they're always going to have to bone up and get these, you know, other fixers for these vaccines, boosters and such. But your body builds its own boosters. Your body knows how to have memory and capabilities like that. So I'm here to tell you, if not preach, that your body building its own immunity will always be stronger than any vaccine. And maybe that's not a popular statement these days, but it's what's been taught to me by medical doctors for the last 20, 30 years until this past year, right? Or past couple of years. Then all of a sudden it's a dangerous statement. But prior to that, it was always the norm to say that the body's ability is always a stronger immunity than anything else. But certainly vaccines are cool. You know, as long as they work, it's beautiful. So what we want for an antigen is the ability to stimulate lymphocytes and to start antibody production. So real quick, what lymphocytes build antibodies? B lymphocytes or T lymphocytes? B, there you go. B for antibody. B cells build antibodies, you got it. And we want them to have this property of reactivity, the ability to react with antibodies. So a complete antigen, one that will make a complete immune response, I should say, will be those that are foreign proteins to the body, which in the case of the COVID vaccine, that was a foreign protein. So instead of the COVID vaccine giving you the foreign protein in the form of the actual virus, you were taught to build the foreign protein with the COVID virus, which is a really weird idea, but it worked for some people. You know, you were given the mRNA messaging coding in your COVID vaccine for your body to build a foreign protein, which is kind of risky, which is why they kept it away since the early 80s, because it was too risky, too volatile of a process. But they found a way to contain it enough to make it tolerable for the body for most people. That was the Pfizer and Moderna COVID vaccines, where you're given the mRNA coding like that in a little phospholipid droplet. The Johnson & Johnson vaccine was a little bit different. You were given the mRNA, but it was inside of a regular virus called an adenovirus, a common cold virus. So your body took in the common cold virus, again, in a lessened form. And then like a Trojan horse, once your body attacked that cold virus, it opened up the coding for this spike protein. And then your body said, look at that spike protein when you fight it. So either way, these were technologies that took, you know, years to make. So on the technological end, it's fabulous, but of course, there's some side effects with anything new coming out. What an incomplete antigen would be is a hapten, what's called a hapten. Incomplete meaning it triggers your immunity, but no antibody. So it wakes you up immune-wise without enough to build an antibody response, but enough to irritate you. So yeah, you get the first and second line defenses coming out, right? The inflammation, the mucus, right? All this crap stuff that we feel when we deal with allergens, all this is allergens. Allergens are incomplete antigens. They're things that irritate us, but not enough to build antibodies. So that's like the threshold between being a complete antigen and an incomplete antigen. Does it build antibodies or not? If it does, it's a complete antigen. If it does not, if it just irritates us, it's a hapten, which would be an incomplete antigen. So if you're making notes on these, this is how you organize your brain. Make a notation to come back. Let this be a study goal. All this active, passive, natural, artificial. Get that encompassed in your head for 15 minutes. Come to a separate story about incomplete and complete antigens. Check that off. Make sure you got that. Talk about it in your head. Talk about it with someone. Right? So attack these little concepts one by one. And I know it's the urge, the tendency to look at every single bullet and wonder if it's going to be a test question. But guys, the way you get through that is understand the basic part of it first, right? And let that sit and marinate in your brain for a week or so. Then come back and maybe get some other little details that make sense to you. Don't on the front end try to memorize every little bullet. It's just not going to happen. Always come back, do it in stages. So remember, the antigen is the bad guy or the bad girl. Your name is Jennifer. You're so Jennifer's near, right? No, okay, that's good. Anyway, so that's the antigen. And you would say, let's just say if we're blowing up on this antigen being the pathogen, that these specific shapes that you're seeing on the antigen, the three different shapes represent what we would call a spike protein. It's a membrane protein. That's flagging the identity of that antigen, right? So in this case, this one has three different ones that will be matched up from three different antibodies. So if this antigen were, a bacteria trying to spread around the body, what do antibodies do? They do exactly what you see in this picture. They basically handcuff them. They hold all the spike proteins. Now that antigen can't do anything. It can't move around. It can't replicate, because you're going to have some perforins coming here and all kinds of stuff drilling in the cell membrane. Or phagocyte is going to come and gobble it up because it's being handcuffed. It's being held. So this is the way that antibodies do their business. They don't kill. They just, distract and play games with the antigens until the killers come. We're going to think of it that way. The red is the proteins on the cell membrane of the antigen. The purple things are the antibodies that match those protein markers. Yeah. They bind and distract. Yep. It'd be like them handcuffing them to the, to the door while the police come, So yeah, antigenic determinants, fancy way of just saying they have certain proteins, certain shapes of proteins. And
sorry.
Yeah. Antigenic determinant is just a fancy name to say certain proteins on their cell membrane. So if you think of, certain plastics, when they're in, like for breast implants, they've got it all figured out mostly now, but early on, breast implants, like in the 70s, were pretty reactive. All the plastics they were using back then were very reactive with women's immune system because of these plastics had certain proteins, that reacted with certain antibodies. And you should know, because the book really doesn't tell you this. It's like a big secret. All antibodies are is a receptor that's been cut loose. Antibodies, we'll see in a picture coming up, all this antibody is, or once was, it was a receptor on a B cell that actively bound with an antigen and was just released into the bloodstream to go find more antigens. So when an antigen meets a B cell, we'll see the pictures of this. An antigen meets a B cell, they bind, right? Now that there's a successful binding from the shape, the B cell now bound, number one, will now be cloning itself. Many of those clones will become plasma cells, which will then release these receptors in purple to be antibodies. That's what we're going to see. And then some of those will become memory cells for a rainy day later on. So they're going to bind, right? Successfully bind, and they're going to clone. Some of those clones become plasma cells. What's plasma mean? Think about it. Plasma is for the blood, right? Where do we release antibodies? In the plasma. So it makes sense that plasma cells build antibodies. So maybe seeing the picture will help. And then there's this MHC complex that we're going to talk about. Basically, it says a set of proteins, which is true, but where do proteins ultimately come from? Who can answer that from AMP1? Yeah, let's further back. There you go, DNA. Coding on your DNA, I think it's chromosome number 6. This is chromosome number 6, for whatever reason, has all the genetic information for all the proteins that make up your cell surface protein sequence identification for your body alone, your unique protein sequence on your cell membranes. Unless you're a twin, then they'll be somewhat identical, right? But everyone's got a unique set of cell surface proteins, which is called the MHC, the histocompatibility complex. So it's a set of genes. It says a set of proteins, but yeah, it's a set of genes in the bigger picture. So the exception for not being this would be identical twins. Otherwise, it's uniquely different. It should say uniquely different to each individual, to be clear. So let me make sense of this on the board, because this is the part that's going to mess with your head. Because even on the handout that I made, it's still possibly not clear. So hopefully we can make it clear. Class 1 MHC. And remember, we're talking about proteins. You can say genes, yes, but we're talking about cell surface proteins. And then class 2. So here's the easy part. Class 1, you can, instead of saying MHC, because as soon as I say that, you probably check out because it's a new term. It just doesn't jive with you, right? And I get that. So just say class 1 proteins. And these are cell surface proteins. So it's no magic. All cells have class 1 MHCs. It doesn't seem that very magical, but this is all cells will have a familiar set of proteins, which means your ID for your cells, basically. Just like I mentioned in my AP1 class, if you fly over St. Louis or fly over New Orleans or LA or New York, right? You're going to see certain buildings, certain landmarks, certain rivers, or whatever. You're going to notice that city for the surface. Just the same as we have a certain identity on our surface of our cells. Just the same as we talked about red blood cells, having a certain antigen in the same way. But we don't call these antigens. We call these proteins. I'm sorry for that, because proteins are antigens. You know, so we can keep it all straight. But in this case, we just call them proteins. Class 2 MHCs, instead of being on all cells, they're going to be on specific cells. Most importantly, on our good old APCs. It's not really possessive, it's just which a B cell can also be having these. A phagocyte, a neutrophil. So basically immune cells, if you want to think of it that way. So these specific immune cells will have a different class of cell surface proteins. Well, here's the magic. They're matched or sensitive to a receptor. So the receptor that matches a class 1 set of proteins is going to be called a CD8 receptor. The receptor that matches a class 2 set or complex of proteins is going to be a CD4 receptor. What makes this hard, guys, is you possibly never heard this before, so your brain naturally wants to shut it out. But honestly, it's not so hard to remember. CD8 goes with class 1. CD4 goes with class 2. What you're missing is you can't see it in your head, so it's hard to hold on to. So you just got to imagine that population of proteins of class 2 only exists on those type of immune cells, whereas this complex of proteins, class 1, is on all your cells. So here's the next step. The CD8 receptor is going to belong to the cytotoxic T cell. And we draw it as a TC with a low C at the end. So a cytotoxic T cell is a big T with a little C next to it. The CD4 receptor will belong to the T helper cell. And that's drawn with a big T, little h. So now we can play with test questions, right? Malaya? She's still giving me the evil eye.
What did you say the CD8 receptor involved in?
Cytotoxic T cell. Yeah, if you want to follow this on the handout, it's written in there. It's just written with a whole bunch of other words too. So in other words, if we're looking at T cells now, there's different T cells, but we're only going to really worry about 2. Okay, the cytotoxic T cell, which is no other word for it. This is the badass, the cytotoxic T cell. Okay, this is what protects all your cells every day from cancer. The cytotoxic T cell is going to have a CD8 receptor, which is again another protein, because everything's proteins, guys. It's going to have another CD8 receptor on its cell membrane that's going to be sensitive to, and notice, class 1 MHC complex on all your cells.
not in class one, not included in all of them.
It's not in the class one, but it recognizes class one.
Yeah. Um,
It's trained to recognize class one group of proteins.
and is that also, okay. Would have to have a vote. Ourselves actually.
No, it can kill alone. It can kill alone. Yeah. It's going to recognize things and kill. Yeah. Straight to the cell. The class 2 MHC will be only on those B cells, like APCs or phagocytes. And it's going to be sensitive to a CD4 receptor on a helper T cell. So match up the, the receptors with the type of complex, and you got it. So this is a nice little passage in the PowerPoint here. These are our two adaptive lymphocytes, B and T cells. This is a great thing to review right here. These 5 communication things. And APCs communicate second line to third line. So remember that an APC can be your regular old macrophage that instead of just ingesting the pathogen, it'll ingest it and then take a piece of that pathogen and bring it up to the cell membrane for viewing, right? It's like taking a criminal to the jail in a sense, and then putting them in a bay window so everyone in town can see the criminal, and then he can get stoned or something. I don't know, maybe in the olden days, probably something stupid like that going on. Yeah, we're going to take a piece of it, bring that protein to the cell membrane. And when a piece of it's on the cell membrane, that's when it's known as it presents the pathogen to another cell, which is going to mount some kind of attack. So these APCs don't respond to specific antigens, they respond to any antigen. That's why they're still considered second line at this point. They're macrophages that work with T cells. We're talking about APCs right here, which stands for antigen presenting cell. These are macrophages, guys, that work with T cells. They digest and present antigen cell fragments to T cells to make them interested and aware. So T cells depend on these APCs for doing their work. They're scouting around the body. This awareness will activate the T cell once found in the antigen. So in other words, it's like a safety net, really. This keeps T cells from going on a rampage of killing without there being an APC in the mediation of all that, right? That's the idea. There's lots of mediation in the immune system. There's lots of mediation in the digestive system with all the enzymes you can see. There was mediation, you could say, even in the clotting. Remember, it took this thing to make that thing happen. That's all considered mediation. And then activated T cells in turn will release chemicals to mount a full defense. So how do you activate a T cell? It's got to bind with the APC, right? It's going to bond with that protein fragment on the APC. That's what activates the T cell. All right. So yeah, come back and tag this. These are great statements, guys. Come back and tag this slide to review that. Really, what we've said now is what we're going to just keep saying. That's kind of all the new data, all the unique data coming at you as far as how everything works. We're just going to now see it work. And once you see the video, you'll get a feel for this happening in so many different ways, you've got to activate these cells by binding. And then once they bind, they start releasing all these cool chemicals that will basically turn on immunity in a bigger way. We're going to turn on more B cells to clone once a T helper cell has bound, right? And we're going to proliferate more T cells once something is bound. It's going to turn on chemicals to activate more T cells. So the T helper, as you can see, is the grand, organizer of the immune system. When a T4 is active, a T helper, I should say, when a T helper is activated, it's going to make more B cells do their thing and more T cells do their thing. But if you don't have T helper cells active, your immune system is only happening on a very low scale. This is the general, as you could say. This is what gets all the troops moving, is that T helper. Very important. What's that? The APC, the class 1 is certain proteins that are on all cell membranes. Let's just say if a virus infects a regular cell, on a regular cell like this, the cytotoxic will notice the viral infection and just kill it right out. Okay. Yes. For class 1. For class 2, which is going to be the bigger part of the immune system, that's the way it happens over there. And I have pictures, if that helps. We're going to see some pictures. Is that cool? What's that? Okay. All right. So for any of these immune cells, B and T, to be up to the job, you could say, they have to go through training. They have to mature. So we know that they're both made, right, in the bone marrow, but where does a T cell mature? In the thymus for T. Where does a B cell mature? It matures in the bone marrow, so it's a B cell. But they're both made in the bone marrow. They just mature in 2 different places. And by maturing, they learn these 2 properties: immunocompetence, which is a test question. Well, don't want to forget to tell you that. Immunocompetence is recognizing self versus non-self. And then self-tolerant. It's like programming a robot. We want to teach the robot not to kill humans. It does not attack self. So first, got to recognize what's not self, and then learn how to not kill what is self. That's important.
Can you just say that the T cells and the B cells, even though they mature in different parts of the body,
Yeah.
they're still both
Made in the bone
in
marrow.
the red bone?
That's right. All blood cells will be made in the bone marrow, no doubt. But these 2 mature in different ways.
This happens to the red bone marrow?
The red bone marrow, that's correct. That's where they're made. From the hemocytoblast, that stem cell, But the T cells happen to migrate to the thymus for maturation. And once they're mature, they might go back to the bone marrow. Or they might go hang out in the spleen. Or they might go hang out in a lymph node. They move around. Same with the B cells. They mature in the bone marrow. Then once they're mature, they may stick around in the bone marrow. Or they may go in the blood. They may go to the lymph nodes. They may go hang out in the spleen. Same lymph organs we learned. So this picture just tells you or shows you what I was just saying. You want to take the time to look at it. They're made in the bone marrow. This is the first picture here. Both B and T, made in the bone marrow. Where does the T cell go to mature? thymus. I don't know why. Just does. Under the hormone guidance of thymosin from the thymus. So what hormones matures T cells? Thymosin. From which gland? The thymus gland. Right. So once it's mature, it could go back to the bone marrow, yeah. But it could also go to the blood or other lymph organs to sit around and wait. So that's all this picture is trying to show you. Or you could say, does the B cell mature in the thymus? Nope. Right? It does not mature in the thymus. Only the T cell does. So they're both born in bone marrow. They mature in two different places. All right. So 3 stages of the life of a B cell. There you go. Born in the red bone marrow, mature in the bone marrow, and then deployed to the lymph nodes or vessels, tissues or organs. So this text is exactly what that picture showed. It's just basically a translation of that image that we just saw. Same with the T cell. It's born in the bone marrow, matured in the thymus, and then can go to the tissues and hang out, waiting for cooperation with the APC to be activated. All right, so more about APCs here. T cells generally don't recognize antigens on their own. In this case, for the T helper, we need the antigen presenting cell to be required. And this is, again, all the types of cells that can be known or purpose themselves as a APC. What they all have in common, guys, is that they're all immune cells, of course, in some way. So dendritic cells are just macrophages that tend to be far away at the periphery, like in the skin. Macrophages are those general neutrophils that just move around the body, you know? They could be known as reticular cells or B cells. They all function as APCs. So at this point, do we have a comfortable picture in our mind what an APC is? They gobble up a pathogen and present a little piece on the cell membrane to attract a T cell, right? A T helper. What was that?
So what they all have in common is their hormone. Are you referring to APC?
No, I said they all have in common is that they're immune cells.
And you're referring to APCs, though, right?
Yeah.
Okay.
All APCs are going to be immune cells. Yeah.
Okay.
Like they have double duty. So again, we're mentioning MHC, complex proteins, acting as an identification tag that labels all your cells. So all cells will have class 1 complex proteins. Only these cells, the APCs and B cells and phagocytes, will have class 2. This is just the same old thing. So would an APC have a class 2 MHC profile? Yes, it would. What would it be sensitive to? CD4 receptors. what has CD4 receptors? T helper cells. So somehow, some way, you're going to need to match up that information. T helper has a CD4 receptor, which is going to be sensitive to a class 2 complex protein sequence. You're just going to have to put that in a little burrito and eat it. Right. It will help when you see this picture or this video that we're going to show. This is like the major heart, you know, of immunity, of adaptive immunity, I should say, where adaptive mixes in with the innate. So there we go. All in red. Cytotoxic cells, cytotoxic T cells, these are the ones that kill outright. They have on their cell membranes, these T cells have CD8 receptors, which are going to be wanting to bind to MHC class 1 proteins. Whereas MHC2, this is kind of displaced. This really shouldn't be here. This HLA, but that applies to both. So don't worry about that being specific to MHC2. Not honestly, if you really want to know a little backup, let's kind of like unhinge for a second. It's getting to be a little intense. An HLA, maybe, I don't know if I should tell you this or not. No, it's important. It is important, but it's not meant to be a confusing thing. MHCs are on any animal. Okay, you could say MHC of a cow, a pig, or rat, it doesn't matter. This is the known terminology for a specific set of proteins for each animal cell around the world, okay, in our creation. But for humans, humans have HLAs. Humans have HLA MHCs. Does that help at all?
No.
Okay, it'll happen.
Does this taste thing happen?
No, it's going to notice an infected cell and go kill it straight out. Yeah. No, it always has to have a receptor, but it can kill outright. It doesn't need any help to kill. It does not need any help to kill. Yeah. Yeah. So HLAs is just the human version of MHC. Can I say that? Okay. MHC is a more global generic term, right? HLA is human. Right. So again, don't you wish scientists could just keep to one term and stick with it? But they always got to have a secondary term. You know, that's what muddies the water. Not going to change it, you know. We're just here to decipher what's already established. So anyway, yeah, we're going to see this over and over again, try to match up the numbers, CD4 with a T helper cell, right? Going to match up with the MHC2 complex. Okay. So here we go. We'll watch the video after this. When an APC presents the antigen to the cell, to the T cell, it activates the T cell. Boy, that sounds like a test question right there. When an APC presents, here's the antigen, right? Here's the T cell floating along. The APC is reaching out, showing the T cell, look at what I just found. This little booger came into our body. What are you going to do about it? The T cell binds to it. We're going to see what's called some co-stimulation, and then activates that T cell. Once that T cell is activated after being bound, it's going to start releasing all these chemicals. It's going to proliferate more More and more and more of everything, basically. Antibodies, plasma cells, bactericidal chemicals. There's a whole bunch of stuff that happens. So it basically takes your chilled out macrophage from floating around, it turns it into an animal that must kill, that must digest as fast as possible. So it kicks everything up a notch, you know? It's like going from a banana pepper to a Carolina Reaper. Kicks things into gear. And guys, think of this. This is adaptive. So the more it happens, the more your body becomes a killing machine. But if you're never activated, you're never become a killing machine, right? That's what I was trying to talk about. If you kill your exposure, then your immune system just takes a backseat, right? And that's what you don't want. So yeah, it's always wise, especially in the medical community, to wash your hands as much as possible, but you're always going to be exposed to something. You can't live in a sterile world. So it's interesting because it seems like pathogens need us and we need them. Isn't that odd? It's hard to admit that, but maybe that's just the way it is. So here's the life of a B cell. It's going to be when it's met, only when it's met with an antigen, by the way, it's going to become a plasma cell, which will then produce antibodies. So you got to know this sequence for the test, I promise you. A B cell will become a plasma cell, which will release antibodies. That is a for sure test question.
Again, please.
Yeah, B cells, when met with an antigen, a bad dude, will become a plasma cell. And it's the plasma cell that releases the antibodies. But here's what the book does. I mean, the picture tells you, but the book doesn't always say it. Let's take this marker, even though it's not a cell. Take this marker as a cell, right? Take the cap as a receptor, right? So this receptor, this black cap, if it's a receptor, the red markers are pathogens, right? That pathogen happens to match and fit right on that receptor quite nicely. Okay, so if the black marker is the B cell, the red marker is the pathogen that happens to fit just right on that black cap receptor. That binding means a lot, right? That good binding, that good tight binding means that B cell will now be activated. Right now that it's activated, it's going to clone itself. So now we got tons of black ones, right? And that's when it becomes a plasma cell, and the plasma cell is going to release this cap as a receptor. This is your antibody. So when the plasma cell releases the antibody, this is the antibody. The same thing that with the receptor. Now cloned by the thousands. So now you got 1,000 caps floating around waiting to find that same antigen somewhere else. Does that make sense at all? So the antibody was the receptor. Does that make sense? That's that Y-shaped thing that we see in those pictures. So it's going to recognize a certain pathogen. attack, and then it's going to form memory against it. So here we go. Let me show you these next few slides, and we'll see a video, then we'll take a break. Whoops. Oh, shoot. Make sure my make sure that stupid video wasn't playing yet. Mess things up. Okay, so in this picture, this is a B cell. Right here, top. Let me blow it up. Immunocompetent B cell has thousands of surface receptors on one antigen. Okay. For one antigen. Fine. So this is our B cell right here. You remember seeing lymphocytes in your first blood lab? This is just one of those lymphocytes that we saw having a big apparent nucleus, right? What we didn't see in those lab pictures is that those lymphocytes have surface receptors, which are these Y-shaped things. That's actually the shape of them. Well, if you look closer at the very, very edge of the Y-shaped receptor is a certain little nook or a specific shape. This is more like a pointy one on the right, a more concave one in the middle here, the purple. The green one has a more etched, like locking key type thing. The red dots are the pathogen. So they're red, they're rounded. So which receptor will they bind with? Not the pointy one, not the ratchet one, but the more concave, smoother inward surface, right? They bind with this receptor. Now that you've bound that red pathogen with that purple little receptor here in that middle lymphocyte, there you go. That's a nice antigen antibody complex, which is what it's called, right? Now we have a successful binding. What happens next? Cloning, right? Cloning. So when it clones, it's going to clone and remember basically that successful binding. And then those plasma cells that will be those clones will release the receptor, this Y thing, and that will be your antibody. So the fact that we clone them means we match the pathogen with the right shape. So now that antibody is going to be going off, circulating in the blood, looking for any pathogen that has that certain shape to match with. So the antibody is the receptor. That's what I'm trying to tell you. It'll circulate through the blood and stick to any of those pathogens, right? The antibody will know. Antibodies will do something different. They're going to you're going to see there's what's called PLAN. They're either going to agglutinate certain pathogens or blow them up, all this kind of stuff. So we're not seeing the T cell in this story yet. No, this is just antibodies doing their thing. Yep. Yeah, you're mixing stories. You're mixing what an APC would do. This is just what where an antibody would come from. Yeah. So after cloning, most will differentiate into plasma cells, and the plasma cells secrete antibodies. There should be a picture coming up that will show this. Here we go. Right there. So see our antigen. Let me blow this up again. So yeah, we're switching gears in this. If I didn't say that, this is looking at B cell proliferation, with antibodies. We're taking a break from APCs at the moment. Where's my mouse, darn it? There we go. Let me use this pointer. Okay, here we go. In the red is our pathogen, our antigen. It's going to successfully bind with this receptor on a B cell, okay? Notice how it says antigen binding to a receptor on a specific B lymphocyte. Everybody good? So these little green, purple, and blue things on the B cell, these are receptors. They're going to be specific for that one pathogen. Once we bind, we proliferate to form a clone with that B cell, right? So some of those clones will become plasma cells. Right here, this is a plasma cell on the left, these three. Other clones will become memory cells. So less of the clones will become memory cells to be tucked away for another day. Most, like good 90% of the clones will become plasma cells. That's what you need to know for the test. Most clones become plasma cells. And it's the plasma cells, and what do? Notice they're looking naked now. What are they missing? They just ejected their receptors, which are now known as the antibodies. That's what the book doesn't tell you. The book should say the damn receptors become the antibodies, but it doesn't say that. So Sliman's book one day will say that. The damn receptors become the antibodies. You could say the cloned receptors. Maybe that's a better word. We'll say that. There we go. Right. And if you're still having trouble, then it's the mother. No, I'm just kidding. We're not going to go that far, right? But maybe if that's what it takes, right? My God, education has changed so much, you know. When I was in school, I was taught by mostly MDs. They don't hold back. Everything they say out of their mouth is one dirty joke after another. It's not pretty. But boy, they get their point across. They can't do that now. Shoot, I remember raising my hand to ask a question, and the professor thought nothing of pointing at me and saying, "My God, are you stupid?" That's how they answer your question. It's a whole different climate back then. I would just say, "Well, yeah, I'm stupid. That's why I'm here." So for me, I got to know this not through words. I got to know it through pictures. So if you go home, draw yourself maybe a picture of the plasma cells being most of the clones. Only a small part of the clones will become memory cells that are tucked away in our lymph nodes for another day, you know? Then those plasma cells, that's why they give rise to antibodies, which you could put parentheses, those damn receptors, right? The cloned receptors. That's the better way to put it. So now, fast forward, right? Now that's today, that's this week. What about 2 months from now? Okay, we get hit with the same antigen, hot damn, but now we're at the ready because we already got all these receptor antibodies sitting tucked up somewhere, don't we? So now we're saying on a second subsequent challenge, a later challenge to the same antigen, look how many more antibodies we can make. Just that lightning fast. Right here. Look how many more clones we get compared to that first exposure. This first exposure took about 5 days to make antibodies, right? That's why you feel like crud for the first 3 to 5 days when you're sick. You know, about the 4th day, you start feeling like you're turning around possibly. Well, on a second exposure, this only happens in a couple of days. Good 48 hours, your antibody titer serum level of antibody is way up there, and you're like Superman, at least against that antibody you are, against that pathogen, I should say. Yeah. Yep. So again, antibodies are all proteins. They're all proteins. Everything is protein based in the immune system, it seems like. And that is why, to be honest, if I want to go back to the story for a second, I think there's beautiful ramifications of the mRNA coding technology that we've come to see now lately. But there's also some scary implications because they could, even if it's not a pharmaceutical company, some rogue lab could produce an mRNA to come into your body and make you produce whatever the hell they want. They can make you produce cancer proteins, right, to kill you off or something. So I mean, it could be a military weapon of some kind, or just no telling. This mRNA stuff is good and bad. It's beautiful technology, but it can be used pretty horribly also. Because when you have something come into your body that can make you produce whatever protein that it says, to me that's pretty dangerous. But it could also fix sickle cell anemia. It could edit your genetic code, which would be really cool. So as long as it's used in the right way, it's beautiful. This is just a step-by-step. That's what your typical antibody structure would look like. We're not going to worry about that for the test. I'm not going to ask you anything other than just knowing the antigen binding site having a certain shape. I think we already said that. Yeah, we're looking at one of the thousands of proteins that your body, would learn how to build. Now, if they could build mRNA technology to help us increase our antibody production, it'd be pretty cool. It's probably already doing that, but just haven't been, released yet.
And know the binding shape.
Binding site is right on the edge there, just like we've seen. Okay, so here's our family of immunoglobulins. This is the fancy name for an antibody. So don't get those confused, guys. Immunogen was the antigen. Immunoglobulin is the antibody. Both are proteins, okay? But the globulin is always going to be the antibody. Immunoglobulin. It's a bigger protein. So here's our family: IgG, IgA, IgM, IgE. Certain one of these are released in certain situations, like IgE for allergies. I would focus more so on the G and A. This is your normal, bigger amount of antibodies produced, G and A. IgG and IgA have the two most highest concentration.
IgG is allergy, right?
E. IgE.
IgE is allergy.
Yeah.
And what is IgA?
Right here.
I mean, is it allergy also?
No. No. I was saying the G and the A globulins are the highest concentration. That's what I was saying to focus on. So as an example, what type of immunity would G and A provide to a baby from the mother? Do y'all remember that? Would that be active or passive? Passive.
Yeah.
Passive, right. Very good.
Is immunogenicity the same as the immunoglobulin?
Immunogenicity is how much of a response you produce. Where are you seeing that? Which slide?
Complete antigen, the two functional properties.
Oh, yeah. Immunogenicity is the ability to provoke immune response. Yeah.
Okay.
Yeah. That's on the antigen side. Okay, so here at this point, I'm before we get into antibody, well, no, we could actually do that real fast, and we'll do the video, and then we'll take a break. So what I'm gonna have you guys do, because it's not gonna be me talking about each little thing. Actually, I want you guys to just take this and run with it at home at some point, because this is the variety, I guess you could say, the parade that B cell antibodies will give. It's known as PLAN, which stands for certain of these terms: precipitation, agglutination, lysis would be the L. Where's lysis? There it is, cell lysis, and then neutralization. So as I said before, antibodies just kind of trick and play with pathogens. Precipitation just makes things stick together, as does agglutination. Neutralization just deactivates those spike proteins so that they can't harm any of our other cells. And then lysis is where we can drill a hole using complements to come in and drill a hole and blow up that pathogen cell. So this is more so things that you can just, I guess, get on your side. I don't want to spend too much time because it really is pretty self-explanatory with the slides that are there. As long as you don't have to know the mechanism of all these, just know the terms, okay? Precipitation, agglutination, recognize these as the PLAN. This is the action plan for antibodies. This is what they do. Know what the acronym stands for. You got it. Plain and simple. So in the case of red blood cells, it's agglutination because it causes red blood cells to clump. In the case of other antigens, it sticks them together so that now they can't move around the body, so now they can be phagocytized, right? You see how all this is enhanced, and then you end up with phagocytosis. So it's all this is a game to basically busy the pathogen. Say, pathogen, no, look right here, look me in the eyes. And they're waiting for another white blood cell to come behind and gobble them up. It's all just to trick the pathogen, to keep it from moving around the body. In the case of complement fixation, here we go with these complements, which is another set of proteins that come in and drill a hole, causing water to come inside the cell and blow it up. So all this is happening at the same time. Let's just get a feel for those. That's not too hard to look at. So there's your 4 words at the bottom. Yep. Yep. So to answer your question, Malaya, this is not where the APC necessarily comes in. You know, this is antibody specific stuff. And again, don't forget your keyword. This is all humoral immunity. You could say something, here's would be a tricky one, etc. Which of the following processes or mechanisms of antibodies is cell-mediated? None of them, because it's all humoral immunity. It's not cell mediated. Cell mediated is T cell, right? Humoral immunity is always B cell. So that's a tough question that really is based on a simple keyword. This is all humoral immunity belonging to the adaptive third line of defense. That's why I said sometimes you got to rewind and go back to the basic stuff when you look at some of these test questions that sound really difficult. And man, you're going to see that all over the MCAT. That's how they get you. They know you don't understand what the hell they're talking about on some of these questions. They just want you to notice, are you keen enough to notice the basics? It's all a game, it seems. Okay, so this is just explaining each of these PLAN attributes there. So it'd be a good idea to read those. This explains the precipitate. I added that word bond in there. It means that we can conglomerate those antibody antigen complexes together. So now we can flush them out with the general immune clearance, which could be saliva, it could be sweat. That's what that immune clearance is for. You just get them out of our body, or puking. That's a form of immune clearance, isn't it? You have one of those terrible viruses, that clean you out. That's your immune clearance working. Your body's saying, let's get that crap out of here. Man, I once ate some, I don't know what it was. It was something at a Mexican restaurant. Holy Jesus. It wasn't here. It was in Mobile, Alabama. And man, I think I threw up literally in one night, like 24 times. It was unreal, gut-wrenching. And after about the 8th time, there was nothing to throw up, but geez, it kept just, wow. Felt like I was turning inside out. I can't even pass by that restaurant now without getting the heaves. I can't go near it. Oh man. Okay. So let's if y'all can I think since we've been on the subject of this APC, I want to go ahead and do this video now, and then we'll take a break after that. This is a B cell with the immunoglobulin receptor, binds to the antigen, right? It'll be in the video now. Brings it in, digests it, and puts out a piece of it, right? Here's the T helper. Okay, so you're going to hear my voice talking because this is a previous lecture of mine. So the volume may be a little bit low. I'll try to turn it up best I can. But it's worth watching because it's a good explanation of everything that we've seen. Digests it and puts out a piece of it, right? Here's the T helper. All right. So now what we've gotten is we've got a B cell that found an antigen, right? Bound to it. Now it's acting as a antigen presenting cell. Okay, it's still a B cell. It's just acting like as a presenting cell. So it's our B cell. Here's the cell surface protein that's got, receptors, you could say. And here's where it's, this is the part that's making it act like an APC because it digested this bacteria, whatever it might be, and it's presenting it to the T helper cell. So we're going to see the T helper bind to it, and it's going to activate itself. Yeah. This is when the T, right. One of the varieties of things that can happen, yes. Yes. And realize that what you're asking is, yes, other B cells are going to be producing antibodies, but we're just looking at this happening now. Yeah. So it happening all at the same time. Yeah. It is confusing. I know, right? And to make it relevant with what's on the board, what type of MHC will this B cell have? That's what the T cell would have. The B cell would have class 2, right? So that's what this is going to be, right? It's going to be sensitized to a CD4 receptor on a T helper cell. That's the whole purpose of us watching this video, so you can finally put it together.
Okay, so
Right.
do that again.
This is the class 2 MHC. That's the pathogen that it's attached to. This is your CD4 receptor on the T helper cell. Okay, so only that CD4 receptor will be able to bind to this class 2 MHC. Okay, the cytotoxic T cell will not be able to bind to this. This is a different protein.
So
Okay,
that's like you said, given the other B cell, that would be the.
that would become the antibody. Is that what you're asking? Yep. Yes. Yeah. It's just another flavor of something happening. Instead of making an antibody, we activate a T cell. Yeah. I think that this statement may help. When we're looking to make antibodies, we're only talking about B cells becoming plasma cells that release antibodies. When we're activating a T cell, we're talking about a B cell acting like an APC that's going to communicate with a T helper. So T helpers have to communicate with APCs. There's no other way around that. Right. And the APC could be a B cell, or it could be a macrophage, right? Right. And a macrophage is a neutrophil that just went on to become more dangerous, right? Okay. So yeah, it took me a while to get all that straight myself. T helper. The only reason that this communication can happen is because T helper cells have what? CD4 receptors, which are specific for class 2 MHCs, right? Which is on the cell surface here. Okay. So now that class 2 MHC has something different on it, doesn't it? Otherwise, it would be ignored by the T cell. You get it? All right. So when this binding happens, that stimulates other chemicals to come out of the T helper, right? And this is where you get the full response from. See these chemicals coming out? Cytokines. Now it's going to trigger the cloning. So because that T cell activated, now it went in turn activated the B cell to do its thing. So T cells, once they're activated, can make other T cells do more, and it can make B cells do more. So we don't have to know all the different interleukins, just know that once that was bound, it triggers the T cell to start activating all the cloning on the B cell. These are the plasma cells. Now it's secreting what? Antibodies. Perfect. Okay. So see, so that's what I wanted you to see out of this video. That scenario, it did help B cell do the antibody story, but it could also help the T cell activate itself more and more. That's why T helpers are. Who said that? That's why T helpers are essential. That's why AIDS kills your immune system, because AIDS kills off the T helper cell. So then you don't get a full immune system. So any old thing, like common cold, the person can have a cold for months and turn into a horrible respiratory infection because their immunity is just kind of shot. So they have to be on so many medications. Yeah, so, cytotoxic T cells get all the glory because they can just kill outright, but without T helper, your immune system is basically nothing because it can't mount the full.
So you said without T helpers, you don't,
Without T helpers, you can't really mount the full antibody response. Yeah. Okay, let's switch to a different video. I hate that it's that low, man. That's really irritating. So what I'm looking forward to is just show you, these are T helper cells. This is the B cell. This is an APC, a macrophage. What's it going to do? Gobble up an antigen. This
So it swallows
macrophage is
antigen.
acting as an APC. Once it brings it in, what's it going to do? Digest it, right?
So that's the
This is just showing
digested
that's on the inside of the cell
antigen.
now. Lysosome
Going to break
takes
it down.
digest it up, break it into fragments. These are the class 2 MHCs, right? They're made from the ribosome, so they're put on the ER and then assembled by the Golgi, all that.
So the rough ER, with the ribosomes is going to make these proteins. And it's going to bring that digested thing to the edge of the cell, like a flat.
Thing you learn in AP1. Either way, class 2 MHCs are coming and they're going to bind to what? The fragment. This is still in the cytoplasm. So now that antigen is going to fit on that class 2 MHC. And where is it headed? To the cell membrane, like a flag, showing that something has invaded it. So the exact same thing happens on the T cell, or excuse me, on the T cytotoxic side, but it just won't be on a macrophage. So now we got class 2 MHC with an antigen fragment. So what's it going to bind to? A T helper. Beautiful. There we go. There's the T helper. There's the CD4 receptor, right? Beautiful. Okay.
What happens to them? What happens to it after that? Does it survive after that, or?
It
This
can.
is a
Sometimes it's
co-stimulation that we haven't
once
talked
the
about yet.
chemicals are released,
It's
it
like a little
stimulates the macrophage
way to
become
check
more
to make
dead.
sure everything is good to go.
And sometimes it can kill itself or just kill everything inside of itself.
So not only does the
So these are what you
CD4
call
match
co-stimulatory factors. We don't have to really learn specifically
these
what these are.
co-stimulators have to work
But it's not just that one binding.
as a second
It's like
match
turning a special key for a nuclear bomb. 2 people have to do it, Co-stimulation factors, cell adhesion factors will occur before it starts mounting a full response. But I'm not holding you guys to know those specifically. Just know that it has to be a co-stimulation before we release all these dangerous chemicals. Okay, so that's
If you're taking anatomy and physiology for the first time, I can guarantee it's not like any course that you've ever taken. And that's why so many...
That's the macrophage doing its thing. So we saw how antibodies are produced, right? Now we're seeing how on macrophage, acting as an APC, binds to the T helper. Okay. Question somewhere?
you answered it.
Okay. So these chemicals that are released are now stimulating other macrophages to become more deadly. So now they're programmed. Now they're programmed to do more of the same thing. So they went from the macrophage now to show you the same thing we already saw with the B cell. The B cell will do what? Have its immunoglobulin receptor, bind to the antigen. What does it do? Brings it in, digests it, now puts it back on the cell membrane and binds to a T helper. Okay, so the more you watch it, the more it's the same old story. I'm just going to flip over to the T cell. All right, so here is any old cell in the body. They're just going to pick a lung cell. So this is not a B cell, it's not a macrophage, it's a tissue cell. All tissue cells have what? Class 1 MHCs, right? So now this tissue cell, right, has been invaded by some virus, right? And this virus is going to be producing different proteins on the inside of the cell. So that's why they call it endogenous, because it's on the inside of the cell.
So this is, we look at the class 1.
Proteins are inside already, because the virus is inside. So now these protein fragments are going to be picked up by a class 1 MHC. So this is inside a regular cell.
That's the rough ER doing
The
its thing.
class 1 MHC is traveling, made by the ribosome, traveling up.
That's a
It's
Golgi
already
vesicle.
bound to the foreign protein from the virus that infected it. It didn't ingest the protein was made inside, okay, from the virus inside.
So yeah, what was different about this, let
Whereas the
me
other
pause
B
it,
cell-
is that instead of ingesting the pathogen, this was the virus that invaded the cell to begin with. The virus started producing these funky proteins inside the cell, inside of our cell, one of our many, many cells. And then because that protein was produced inside of our cell, that's why it's called endogenous. So then we just take a piece of that protein and bring it to the cell surface. So we didn't ingest anything, the virus came on in on its own.
Cell thing was the exogenous. So now, what do we have? This is a cytotoxic T cell with what kind of receptor? CD8. Beautiful. This is our infected cell with the class 1 MHC with an antigen bound to it, an antigen fragment. There's our CD8 receptor. There we go. So now what's going to happen? It's going to release chemicals now that it's bound. We got co-stimulation factors going on so we don't kill it by mistake. You know, it's just an extra.
So this is the
security, let's say.
cytotoxic T cell just killing outright.
And it'll just cause lysis of the cell. Just breaks up. Okay. So we don't have time
So
for
real
this next
quick,
one, but
this is
I
what went unsaid, shame on me. Notice we didn't really kill the virus. We killed the cell that the virus had invaded. So the virus goes on possibly to do something else. That's why you can't really, I mean, you can deactivate viruses, but you can't truly kill them outright to get rid of them completely. Yes, there are some drugs that can, but you know, viruses go on to produce problems all the time. That's why it's tough to wrangle up a virus. We can take our break. We'll come back and watch the little, there's not that much left.
So I don't say virus, when I'm thinking about a virus, it's normal to have to take its course throughout the body. There's nothing that can really kill all.
Yeah, the best we can do to a virus is contain it, so it can't spread anymore. Right.
So-
That's why it takes 5 days, 6 days to contain it.
And so eventually when you are no longer high viral,
Yeah,
whatever.
you deactivate the virus, you take the dangerous capabilities away, which didn't necessarily eradicate it.
So when the body is breaking down that viral.
Yeah, the protein, so that's what makes the virus dangerous. It's a little spike protein that it creates. It irritates, it, you know? Yeah.
Is actually pretty nice because the guy goes through some different, it's clearer pictures that you can see. And he kind of explains it more of in a lecture type of a way. So if you want to watch that one,
Actual video is on,
that'd be good too.
but I'm just showing you a piece of it.
I'll show you what's going on a little bit. The T helper, let me go back up here. This is the helper T cell right here, right? Remember the language that we did.
Helper T cell.
T cells communicate with what?
APC up here.
APCs or B cells. Helper T
Helper
cells
T
are
cell.
going to ignore
APC.
regular body cells, aren't they? And they're going to want to do what? Look for B cells and APCs, right? That have the class 2, right? So that's
The
what we're
regular
showing
body
you right
cell
there.
class 1, APC will have class
This is a infected, right? This is an antigen right here. The helper cell doesn't go to the antigen, it goes to the APC, right? That's already been infected with the antigen. So that's the difference.
The purpose of this picture is to show you that the T helper will not go to a straight generic infected cell. The T helper has to communicate with what? The APC. That's the point. Okay? The T helper cell will have to communicate with an APC. This body cell that is infected, that needs help, what will it bind with? The cytotoxic T cell, right? Okay. So your regular body cells, all your body cells have class 1 protein sequence. So what receptor mixes with class 1? Very good. The CD8, which is found only on cytotoxic T cells. So it's the T cell that has the CD8 receptor, right? It's the infected cell that has the class 1 MHC. Maybe that's what's been confusing. It took me a while to get that too. But that's how you match up the terminology. What's that? I can't tell who's talking. Oh, how long? Oh, about 50 years. Since 2000. Yeah. I've had several lifetimes, it seems. Oh my goodness. But I have found no matter what book you read, they really don't just step by step spell it out. They just kind of blast it out there. So it takes really doing this slowly. That's why I wanted to actually go through the video with you, because I think this is the biggest. What does act straight on the antigen would be something like a natural killer. All right, that's where you got to kind of know what each cell does. The helper T cell goes to the APC or the B cell. The cytotoxic T cell goes for the infected body cell. Natural killer cells and general macrophages will go for the actual antigen. Does that make sense?
Yeah.
Okay, so it's written on the board, right? And we've talked about what happens. So now the cytotoxic T cell, the only confusing part of this, it has it binding with this. I wouldn't go for that. So just ignore him talking about the cytotoxic binding here. The cytotoxic cell is going to do what? It's going to look for. This can happen, by the way, but what I'm going to put on the test for you is to make it more straight line, that the cytotoxics will go straight to the body cell, okay? And the helper will go with the APC. That's what I'm testing you on, okay? That's my promise to you. Uninfected cell. So let me show you where that happens. Right there. This is an infected cell. So that cytotoxic cell is going to bind to that, because what kind of MHC is on this one?
I have to stop and tell you guys about Lecturio. Lectur.
Cytotoxic will go through the same kind of cloning that the B cell did. Some of those cells will become memory T cells. Some of them will become effector cytotoxic cells that will kill this one straight out. See, it's binding to it, and that kills it. Okay? So just give that a watch if you can.
Okay.
All right, so let's get back here.
Okay, y'all. That is that. Let me go ahead and close that so it doesn't come back and haunt me. So that's on the YouTube channel. You want to look under playlists, and it's under the immune system channel if you've ever been here before. All right. So looking back at antibodies, we're just saying that on a second exposure, it takes within 24 hours, you start building much more antibodies, higher levels than your primary response, because you've got those memory cells at your favor now. So on a first encounter between the antigen and the B cell, usually in a lymph node or something like that, your antigen will bind, as we said, right? Once it binds, then we can start triggering cloning, right? This is what you know, test. Once we bind, we get cloning. That triggers the cloning. It's not going to clone unless it's a successful lock and key binding. Then the B cell will grow and clone itself to become mostly plasma cells that will then secrete antibodies. There's that same sequence we saw before. B cells, plasma cells, antibodies. Please know those three in a row. And then a few of the clones become memory cells that will tuck away in the lymph nodes again, or in the spleen. So we kind of saw some of this before. IgM is one of the most apparent ones here, and IgA. So here we go. This is the graph I was going to tell you. This is our first exposure. First exposure to the antigen on day 0. It took about roughly 5 days to start building some decent antibodies, right? But look on the second exposure to the same antigen. Basically 2 days later, really before that, look how much more antibody production there is on a second exposure to the same antigen. So that's the kind of test question you would expect. Will there be a larger or smaller antibody response on a second exposure? Much larger. Yeah, that's what you want to know. And a quicker timing for it to get there. Look, it took from 7 to 10 days, right? 3 days to get at your top peak antibody. It took really just a day or so to get your top peak antibodies up there. Now, the pink represents a new exposure to a brand new antigen, so that's why it's still low. This is happening all day long. You know, you get exposed to all kinds of crap every single day. So there we go. Review on this. Helper T cells will recognize class 2 MHC antigen complexes. One more time. And that's just another picture to show the same old thing. We gobble up the antigen, then we present a piece of it on the surface, right there. This is the receptor up here at the top of the picture. This little red piece is the class 2 MHC. Then this is where the T cell binds to it and makes the B cells go crazy. That's what also triggers the cloning. So the difference is, yeah, the B cell can do its own cloning by itself, but it happens more and more with the help of the T helper. Does that make sense? Because I think that's what Malaya was asking. She was seeing why it happens in both ways.
The B cell can do it all on its own, yes. But the helper cell just makes it happen even more, okay? So that's why we see the B cell acting like an APC to join in with the T cell, which triggers more cloning. So test question, what's the best way to get the most cloning, the most antibodies? B cell proliferation or MHC, MHC APC binding? The MHC APC binding with the T cell is where we get more and more and more clones, right? That T helper is the key. T helper is like the general that produces all the troops to come out in a bigger way.
You said the T cell help, the B cell can clone by itself, but the T cell is what? Helps it clone.
More,
Better.
more better. Yep.
I'm sorry.
No, I said it like that. Just to be funny. I wasn't trying to mess with you. It's just me being stupid. The APC, a B cell acting as an APC, activating a T cell. A T helper, I should say. So the key to that is T helper. What's the best way to produce antibodies? The help of a T helper. That's the best way. All right, so this is a play-by-play. This is a antigen. Look at that little sucker. We ingest it, right? We merge it with a lysosome. We digest some material. And here's one of the proteins we happen to build all day long. If we're on a APC, this is a plasma membrane of an APC, so this could be a macrophage, right? What are we going to produce all day, every day? Proteins. One of those proteins is going to be class 2. So this is going to bind with the class 2. And where is it going to go? A piece of it's going to fragment and bind to that class 2. Now we're waving like a flag. We're an APC waving a flag with an antigen. What's going to happen? We're going to attract a T helper, right? That helper T cell will have a CD4 receptor, and it's going to bind to that sucker right there, and you know what happens next. That's going to activate the T cell to release chemicals, right? Which will increase more T cells and will increase more B cells. That's why the T helper is so famous. It helps both sides of your adaptive immunity. So there's just a summary, but we're really only picking on these top two, the helper T and the cytotoxic. So just match up what receptor is going to be found on each cell. The cytotoxic will have the CD8. The helper will have the CD4. So when you learn proteins in AMP1, you had no idea. You'd be killing these proteins to death in AMP2. They're all there. So there's some in the red. I've kind of shown you what these what these T cells will do. It's been shown through research that the memory cells for the T family, this is what's at the weakest in vaccines. Vaccines don't produce healthy, good amounts of memory T cell immunity. They produce decent amounts of helper T cell immunity, which is why the vaccines are, useful. But what they don't do is produce a healthy memory T cell immunity. A natural exposure would be the way to do that. And that research is, 50 years old. So it's nothing I'm just saying now to cause trouble. It's just what's been written in the books forever. Again, in the red,
Response is controlled by co-stimulation. In order for a T cell to be activated, notice the title of the slide. Notice what's in bold. They must first bind. And then what's important? Co-stimulation. So make this slide nice and short. T cell activation requires co-stimulation. Bam, that's it. Move on. Once bound and activated, what does it do? Clones, just like the B cells will do. Some signals will stimulate cytotoxic T cells, like what we saw in that video just now. Other signals will stimulate production of B cells, which will then turn into what? Plasma cells, which will then turn into releasing antibodies. There you go. So, man, this slide in the red, in the black bold, a good, nice little summary of what to expect. So yeah, it is a stretch. I get it. Trust me, I get it. To match up these receptors with the right protein complex. If this is your first time seeing this, hopefully you can chew on it for a while and get a little bit of it, you know? And then seeing highlights like this are going to be real important to bring everything together. The bigger story, how important T cell stimulation is going to be for the T helper. It can never be, understated or overstated, I should say. The T helper is where it's at. You got to have T helper cells. That's why HIV/AIDS condition is crucially bad for the human body. It makes T helper cells kill each other. Okay, so that's just another look at a T cell being activated by binding, and then it releases its own little memory cells, just like we saw in the B cell. Same old story. Just give these pictures a look. You can learn a lot from a picture if you're patient enough. It's it's the easiest thing to want to skip over all the small writing, but if you can stomach it out, I promise you could probably learn a lot just from one picture. All right, so this is another another cascade which we don't have to know, but it's just showing you how T cells will proliferate, right, into more helper cells, which will then activate more B cells, and then some of the T cells will go on to proliferate more T cells. So that T helper here at the beginning, matching with an APC, look at that. Right there, look how much it did. It proliferated B and T cell, full spectrum immunity. That's why the T helpers are where it's at. The CD8s, they also proliferate, but they're more so cytotoxic, direct, kill and shoots. They're not as widespread as the T helper would be. Okay, I think we're getting close, guys. Real close. So some of this is what we've already talked about. This is a brief look at the class 1. So this would be on all cells, and if I didn't mean to confuse you, but class 1 is all cells, which means these have class 1 also. But these specific cells have class 1 and class 2. Okay, that's what makes them unique. So yeah, this is your average cell all around the body, no matter what cell it is, right? It will have a class 1. If a virus invades it, what's going to happen to that virus? It starts producing its nasty little proteins, which are going to bind to our class 1 MHC. And this is considered endogenous because the protein is built on the inside. It was not a protein that came in. The protein was built by that virus hacking our nucleus.
to now build a new protein. Right? So that's what viruses do. They build their own nasty little proteins. And that's why we feel sick. So now we bring a piece of those proteins to the surface, and then what cell is going to be receptive to a class 1 MHC? What was that? The cytotoxic T cell, you got it. Right. So there you go. The cytotoxic T cell will now target a class 1 MHC and do its best to kill it straight out. Okay. Good. The last part of this chapter is talking about some hypersensitivities. I think I'm going to just let you read on those yourself. It's kind of self-explanatory. A type 1 would be a very, very fast response type of allergy, immediate hypersensitivity, whereas a type 4 is a more delayed response, about 2 weeks. Oh, excuse me, a couple of days or so, mostly after. So you can read on that yourself, because most of this is just, it doesn't really need any explanation, it just is what it is. So I think that's the end of the PowerPoint, guys. Look for some of these immunodeficiency things, just read through that, look for the bold words. All right. Happy Valentine's to you. We'll pick up with endocrine on Thursday, and we'll have our lab for endocrine on Thursday as well. No, the quiz is on lymphatic,
Okay.
but we will have endocrine case studies to do for Thursday.